Thank you. I'm really glad that there was a question on how we can use genomic information to inform drug discovery or repurposing, because that's what I'm going to give you exactly here. Depression affects over 200 million people globally, and new treatments are much needed. However, making a new drug from scratch is very time-consuming and very expensive. And despite all the time and money investment, 90% of drugs actually fail during clinical trials due to safety or efficacy-related reasons. In comparison, drug repurposing represents a cost-effective and time-efficient alternative method for developing new treatments for depression. And one type of drugs that has been proposed as a repurposing candidate for treating depression is the statins. Statins are inhibitors of a protein called HMG-CR, and by inhibiting HMG-CR, they can effectively lower LDL cholesterol levels and are widely used to treat cardiovascular diseases. Now, some small-scale randomized control trials have shown that statins could lead to further improvement in depressive symptoms when used in conjunction with an antidepressant. However, the overall evidence from observational studies and other trials on whether or not statins have any effect on depression, beneficial or adverse, remains unclear. So here, we tackle this question using a genomics approach, as many statistical genomic methods, such as the Mendelian randomization approach that we use in this project, are less prone to reverse causation and unmeasured confounded bias, which are major limitations of observational studies. And in this project, we mainly wanted to investigate whether statins were likely to have any beneficial effects in treating depression, and if so, what the underlying mechanisms of action were. So the first genomic approach we used was transcriptomic signature matching, and it's very similar to fingerprint matching, but for drugs. So in this case, the fingerprint of a drug is the gene expression changes that it induces in human cells, also called its gene expression signature or transcriptomic signature. And just like how the fingerprint of a person can indicate their identity, the gene expression signature of a drug can also provide unique insights into its pharmacological actions. And the underlying hypothesis of transcriptomic signature matching is that if two drugs have very similar gene expression signatures, then they're likely to have similar pharmacological effects. With this hypothesis in mind, we leveraged the drug signatures from the connectivity map database, or CMAP. So how does CMAP work? So on the figure here, I have, that looks funny. But basically, how the CMAP works is that the CMAP team first expose some human cells to the drugs of interest. And in this case, I'm using statins as an example. And after a period of exposure, they then profile the gene expression changes induced by this drug exposure and basically identify the genes whose expression was either upregulated or downregulated by this gene, by this drug. And these differentially expressed genes would then constitute the drug signature of statins. So we retrieve these signatures from the connectivity map database. And what you can't see here, but what I'm trying to show here, is that we compared it against the hundreds of thousands of other drug signatures in the connectivity map database. And what the connectivity map algorithm then does is to compute a tau score, which is a measurement of how similar or dissimilar a pair of drug signatures are. And a highly positive tau score will indicate high similarities between a pair of drug signatures. So using this approach, that looks funny again, but using this approach, you just have to take my word for it. We identified a number of drugs whose signatures were highly similar to those of statins. And amongst those highly similar drug signatures, we found the signatures of 12 antidepressants in total. And in fact, a chi-square test revealed a significant enrichment of antidepressants amongst the highly similar drug signatures. And what that means is that as a drug class, antidepressants actually show more similarities to statins than expected by random chance. So we're quite encouraged by these results, and we then perform further analysis on the top 5 antidepressants that show the highest similarity to statins. There you go. Okay, so what we did here was to try and understand the biological processes that contribute to this high similarity. And basically, for each pair of the statin and antidepressant signatures, we identified the genes that were perturbed in the same direction. And these genes are highlighted by green dots on this simplified illustration. So basically, they are the genes that are either upregulated by both antidepressants or statins, or they were downregulated by both drug classes. We performed functional enrichment analysis on these genes and found that these genes that were perturbed in the same direction by statins and antidepressants were functionally related to diverse biological processes, including various innate and adaptive processes, as well as some inflammatory processes. So these results were very exciting because they corroborated previous experimental evidence on an immunomodulatory effect of statins, and they also showed that the high similarity that we saw between the statin and antidepressant signatures were at least partly conferred by a concordant impact on immune pathways. So encouraged by those results, we then proceeded with a second genomic approach called Mendelian randomization, which Professor Yang touched on before. It's often referred to as nature's randomized control trial, and it's a statistical method that uses genetic information to infer the association between a modifiable exposure and an outcome. So how does it work? So here I've shown the setup of a randomized control trial for the purpose of comparison. So in a typical trial, participants are randomly allocated into a treatment or a control group, and individuals in the treatment group would then get the treatment of interest, in this case, statins. And as I mentioned before, statins are inhibitors of the HMG-CR protein. So as a result, individuals in the treatment group will then have a lower level of HMG-CR protein activity compared to individuals in the control group. And at the end of the trial, we can compare the outcomes between the treatment and the control group. And here I'm using coronary artery disease risk as an example. And if there is an improvement in the treatment group, then the conclusion would be that statins have a beneficial effect in reducing CAD risk. So in the setup of a Mendelian randomization method, individuals were actually grouped based on the genetic information that they were born with. So this is also called genotypes, which is a fancy way of saying the version of the genetic variants that they inherited from their parents, determined by the random allocation of genetic variants. So as shown on this figure here, individuals with genotype B, highlighted in yellow, they were born with a version of the HMG-CR gene that predisposed them to naturally expressing a lower level of the HMG-CR gene expression. And here we assume that a lower level of HMG-CR gene expression is a good proxy for lower protein activity, and we did perform some sanity checks to make sure that the assumption held. So here, these individuals would be our proxy for a treatment group in a trial, as if these individuals have been taking statins from birth. And on the other hand, individuals with genotype A, highlighted in blue, they were born with a different version of the HMG-CR gene, and they naturally express a higher level of the HMG-CR gene compared to individuals with genotype B. And they would be our proxy for the control group in the trial. And again, we can compare the outcomes between these two genotypes and infer an association between genetically predicted HMG-CR inhibition and our outcome of interest. So using this approach, we investigated the association between genetically predicted HMG-CR inhibition, proxy using ETLs, which are genetic variants associated with the HMG-CR gene expression, and we assessed their associations with diverse depression-related outcomes, as well as some blood biomarkers because of the findings that we observed from the connectivity map analysis. So here comes the results. So unfortunately, we didn't see any association between genetically predicted HMG-CR inhibition and depression risk, but we did see a weak association with a reduction in worrying, which is a symptom of depression. But the most exciting results to us was this extensive and strong associations that we saw between genetically predicted HMG-CR inhibition and platelets and monocytes. And both of these cell types have previously been linked with depression, where platelets are a major reservoir of serotonin, and monocytes have also been observed to be activated in patients with depression. So in conclusion, hopefully I've shown that genomics is a powerful tool that we can use to assess drug repurposing opportunities. And in this particular genomic project, we show that statins could potentially induce an antidepressive effect by influencing the immune system, which has been shown to go haywire in certain patients of depression. And our findings also warrant further clinical trials looking at the antidepressive effects of statins. And lastly, we also highlight specific blood biomarkers that should be considered in downstream trials, particularly to stratify patients and identify those who are most likely to respond to treatment. In the end, I would like to thank the co-authors on this project, especially my supervisor, Dr. Sonia Shah. And if you're interested, please check out our publication there. The QR code will take you there. And there's also a YouTube video on the fingerprint matching approach for drugs. And lastly, I would like to thank the funders and all of you for listening. Thank you.
Thank you so much for the talk. I think we've got several people talk. Do you want to start the question?
It has Jiayue from Westlake University. A few quick questions. In the Mendelian randomization analysis, did you use just a cisNAP or you done that genome-wide? Have you looked at different cell types or tissues and the pQTL?
Yeah, those are really great questions. So yeah, we only use cis EQTLs, and we performed the primary analysis using EQTLs from our whole blood because that was the biggest data set we had. But we also replicated the findings using brain-specific EQTLs as well.
And other protein QTL, have you looked at that?
So unfortunately, we couldn't find any available pQTLs that are strong enough to proxy for HMGCR protein expression, but that's something that we hope to do in our future analysis. Thank you.
Hi, I have a couple of questions. First question, with the statins, so a lot of the statins don't cross the blood-brain barrier, right?
Yeah.
So I guess, do you see like a difference between the ones that cross it and the ones that don't? And how are you accounting for this, right?
Yeah, so that's an excellent question. So there are a few statin compounds available on the market. And the reason why we see conflicting results from observational studies and trials, I think, is partly because those studies looked at different statin compounds, and the variability in the biochemical properties could potentially contribute to the variations in findings. So with the genomic approach that we have here, we proxy for the primary target, the on-target effect of statins. So that's the primary effect of all statin compounds that they all share. So our results are less, I guess, prone to this between compound variation.
Right. And then I guess the other point, CMAP, right, is like based on non-brain cells. Right? So, I mean, it's just kind of hard to imagine because a neuron is very, very different from, I don't know what they use, I'm guessing fibroblasts or something. So what do you do about that bit?
Yeah, so that's another excellent question that I didn't have time to address. So we performed the primary CMAP analysis in HA1E cell lines, which are of kidney origin, and I know that kidneys are not the same as brains. So we did perform replication analysis using a neuronal cell line. And the reason why we started off with a known brain cell line is because that's the cell line that the statin compound, the different statin compounds, show the highest concordance in. So that's why we performed the primary analysis there. But we did perform replication analysis in a brain cell line. And if you're interested, please check out our publication.